be aware that everybody, noises, farting, sneezing, will all be recorded as of now.
And can you put your face down? We don't want to see the speaker there on the screen.
No, you haven't got a choice.
Is this all?
It is, indeed. Everybody can make sure they sign the book as well, and that would be good. Acceptance is the best policy. It will. I can make it smaller if it upsets you, Emma. There you go. Is that better? Okay, look, really I just wanted to try and go through as quick as I can, and a lot of the slides that I'm going to present are actually the slides that were presented by the guys from Oxford Nanopore, so they're not actually my slides, they're actually pinched from his presentation, but I thought it was the best way to go through that particular presentation. I've put a few pictures in just to keep it happy for those of you that might fall asleep. At least you've got some nice pictures to look at. This is actually, those of you who know Oxford, River Thames flows through Oxford, and there's lots of little places where you can go punting on the river. It's a well-known tradition in that neck of the woods. And there's some beautiful pubs, as you can see here, actually sitting right on the side of the River Thames, which is actually quite nice. Not that I got a great deal of time to go and actually do that. The conference itself had lots of good points, and there's some kind of boring points. The interesting thing from the point of view of PacBio was that they are coming out with a HLA typing kit, and that kit will allow fully phased class 1 and class 2 typing on their machine. This is a single molecule platform. Unfortunately, while the actual typing is not too expensive to buy the machine, it's about $700,000 to $800,000, so it's unlikely that that will be coming here anytime soon. But Roche is coming out with a smaller version, which will be only $750,000. Now, I have no idea what the figure is, but knowing Roche, it won't be cheap. And if anybody's interested in that, I've got a pamphlet available upstairs. I didn't bring it down with me, but basically it's just they're able to sequence right across class 1 and class 2 in a single product, so you don't have all this issue with not understanding which phase it is. So the alleles are really well defined and sort of 99.99% accurate in the sequencing. So it's actually very good. It's just expensive. The infrastructure is expensive to get. There was some interesting stuff on the proposal. It's actually happening to reconstruct the first eukaryotic genome from basically chemical bits, a bit like they did with poliovirus. Actually, so we can say that I created life purely from synthetic bits, and that's happening. And if anybody's interested, the link is there. So really want to, actually I'll just go back to that previous slide. This is actually where I stayed at Oxford. This is Lady Margaret Hall, which is all very posh and very nice. These are all rooms, all these areas up here are all actually rooms that you can stay in, but I didn't actually stay in the original. I was in something that was built in, I think, the 1960s, which really is like a sore thumb in the whole place, which they never take pictures of because it's hideous compared to the rest of it. But the rest of it is actually very nice. And I just noticed that my picture's gone, which will please Abba no end. I don't know why it's gone. Alt-tab, it's come back. Oh, there you go, look. Who said Alt-tab? Oh, well done, Ian. This picture up in the top is the Department of Zoology at Oxford University, is actually where the conference was actually held, so all the talks were actually in that building. And Lady Margaret Hall is actually, if you imagine, up the road there, there's a big park, and it's sort of through the big park. So really what I want to go through is this today is really what Oxford presented. There's loads of other talks, and I'll do those another time, but really everybody was really there to hear what Oxford were actually doing, because the MinION Early Access program had been released in February, and some people had already started generating data, others hasn't. Some people couldn't get into the program in the first round. We didn't. We got in the second round. But everybody's really interested in what's happening, where it was going, because they hadn't presented anything since AGBT, right, in 2012. Really nothing has happened. So everybody's really interested. So this was, the place was packed. It was the only session in the whole conference where every bit of available space had somebody in it. Even the steps and the back of the lecture theatre was just covered with people. So that for those of you who don't know Oxford Nanopore, it started as a fairly small company, and it's been, several people have tried to take it over, including Roche. Illumina have a part share in it, I understand, but it's only a very minor share. But they are largely an independent company, and the CEO is actually an Indian man. So a little bit of snippet information I didn't know. So what I'll do is I'll open the slide set rather than go through my notes, because they're actually all in this slide set, and really I'll just go through page by page. I don't know if I can I zoom this out at all? Get rid of this maybe. No.
That's the PDF.
Yeah.
Click on the tools menu and shoot the side button.
Aha, that worked. That'll do. That's no, looks like 125 maybe. No, it's good. 100%. Okay, I've disappeared off screen, but look, I'm back. Don't worry. don't want to keep Abba unhappy. Okay, so I'll have to go down page by page. So he started with a company overview about how many employees they had and how many have gone up over the next time, how much they've had invested, how good their board is, and what intellectual property they kind of had. And then he went on to talk about how the whole process came about. So he went back to sort of history in the beginning, and this was the first time that this process was ever thought of, and this was a book. That's actually the lab book of these two guys, Dreamer and Branton, and it was about 20 years ago where they thought of actually using some sort of hole or a pore where DNA would pass through and interrupt the path of basically an ionic stream, which is obviously a voltage charge across a liquid solution. And as the DNA goes through the pore, it sort of blocks it up, a bit like, chicken pieces blocking up your sink at home, stopping the water flow. The same thing sort of happens here, that stops the electric current flow, or the voltage changes, and you can detect a current change as each base goes through it. It occludes that pore by different amounts, and less ions or more ions flow, and you can do an interrogation. And he envisaged that you would get this nice, if a C's flowing through, you would get a drop of this much of G or T and blah blah blah. So that was sort of the principle behind how this type of sequencing might work. And next time when I do a presentation, I'll talk to you about another presentation that was given. Actually, I think it was the one after this or two after this, on a new type of sequencing. Just when I thought there is no other way you can possibly sequence DNA, somebody came up with another way of sequencing DNA, and it was actually really very good. So I'll take you through that one next time. He went, as I said, back through history and talked about, when MinION was first envisaged and when it was first presented, and the AGBT conference, which was here in 2012, where everybody got really excited because it was the first time the MinION, and all of you seen the MinION that's sitting on my desk? I know Ian's seen it, some of you other guys have. and you'll see a picture of it in a minute. It's only about this big, tiny little DNA sequencer. To make, the guy told me it's about $250 to actually make all the components that go into it, and that can sequence as much as more or less as much as our Flex can, which was 750 grams. So it's amazing how that technology is changing and miniaturizing and becoming smaller. But that was the first time it was presented at AGBT in 2012, and very little was heard of until really the later stages of 2013, when they released the MinION Access project, which was the early access to people that could actually do some work. And that's a picture of the MinION. This is what it looks like. It's a little USB that sticks into a laptop, and I literally mean a laptop. That's all you need to run this. And it's got to be a fairly decent laptop, but it's not anything like any of the computing stuff that we've needed to run a sequencer in the past. And the reason that they have been able to do this is some changes to what's called it's the ASIC. Now, I'm not sure what ASIC actually stands for, but in electronic, there you go, right there, first line should just open up. But what I know about it is that it's basically in the electronic guts of the thing, and that they had to make some major changes to this ASIC component because there was nothing that could actually keep up with the pore. The pore was pulling DNA through so fast that the interpretation of the signals, the electronics just couldn't keep up. So they had to make some changes to the ASIC, which allowed the electronics to run faster so they could actually capture the information, but they also had to make some changes to the actual pores to slow the DNA going down, because we just currently can't compute at that speed. Hopefully it will come, and it's good for the motors on these things because it means that there is a huge amount that can be gained in the future as the process, the electronics catch up with the biological process of passing DNA through a pore. So, as I'm sure you're all aware, a nanopore is just a very small hole, like a mouse's ear. Ionic current flows through that pore, as I discussed earlier, and basically you introduce your analyte in, which blocks the pore up and reduces that flow. So you see a block basically in the flow, depending on the size of that thing that blocks the pore up, essentially. Okay, that's freaky. Okay, and he did say he'd explain this so often he wasn't going to explain it again. So how do we sequence DNA using this type of pore? And it's actually not as easy as one might think, and it's certainly not as easy as you throw the DNA through the hole and it can interrogate every single base. It actually can't do that. It is just way too fast, and the pore is too deep to interrogate just one base at a time. And really the difficulty they've had with this has largely been associated with understanding what the changes in current mean when the bases are actually going through that pore. And that's what's caused a lot of the delay in being able to understand the sequence that's actually going through the pore at any one time. So what they do basically is they take a piece of DNA, and they, at the moment, they attach this, what they call a motor protein. Now the motor protein is completely not what it says it is. The motor protein is not a motor. It actually is a brake. And the reason they have to put the brake in is because I said the DNA goes through that pore so fast that they need to slow it down to actually be able to interpret what's happening at any one time. So that ASIC circuit needs the time to actually detect what's happening electronically during the time that the bases are actually in the pore. And to cut a long story short, they can't do it. You cannot interpret one base in a pore. It just can't be done. So what they actually do is they interpret I think it's between 4 and 6 bases in the pore, and it depends on the type of pore they're using. But essentially it's a 6-base read at any one time. And so that makes it very, very tricky to interpret these electronic signals that are coming off based on 6 bases that might be sitting in the pore. And that's where a lot of the error at the moment is being introduced into the sequence from these types of devices. And that's basically an understanding what they call the word size, so, or what that word actually means. So there's a length to the word, and there's actually an alphabet to the word. In the DNA case, it's either T, C, G, or A, but in what order is that happening in that word? And the ionic signal that you get is different for each of those bases over a 4 to 6 base stretch. So that's where a lot of the difficulty is at the moment in interpreting that signal. But essentially what happens is they read both strands of the DNA, and what's not immediately obvious here, but I think is on some of the latter slides, is the DNA. Maybe if I just go forward, I wonder if they've got it. Did he have a picture? Yes, this one's probably a better picture. They actually tether the DNA to the membrane that the pore floats around in. And you can see here, this is the membrane that then there's a single pore in each well. And perhaps if I just go back to probably here, you can see here, this is basically what each well on the MinION actually looks like, and there's 512 wells on each MinION chip, and in each of those wells there is one pore. And that pore floats around in a lipid membrane, and that lipid membrane basically is what you see here. So this is the lipid membrane that it's floating around in. And so each of these wells, if you like, is able to sample one molecule of DNA at any one time. Now the rate with which that DNA moves through that pore is extremely fast. So once it's finished one strand, it can just start on another one. But only one strand can occupy that pore at any one time. So each of the wells can have thousands, millions of DNA pieces in there, but only one of those is being looked at any one time as it pops through the pore. When it's popped through the pore completely, it can start on another one. And it just keeps feeding them through. And that's why you can, this technology is what's called run to need. And so you can basically keep running it until you've got as much data as you need. So if you don't need a huge amount, you can stop it quickly, but if you need more coverage, you just keep sequencing until, and you can also reload it. So you can stop it and add more to it and run some more if you like. Although there are 512, I think, individual wells, not all of those wells on every chip are fully functional. And there's a period of, and you validate each chip for the number of active wells. So on average at the moment, I think they're getting about 300 to 400 active wells. So only of those 512 that are supposed to have a pore in and be active, only at the moment the technology is only able to deliver around about 300 to 400 of those as active wells. But still quite a significant amount. And that's going to improve over time, obviously, but that's really where they're stuck at the moment. And one of the things they found that improves the efficiency of sequencing is if they tether the DNA to the membrane. And I'm not sure how they do that. I'm not sure he actually did say it, maybe proprietary. But they tether the DNA molecules to the surface. And the reason they do that, if you can see here, if you've got a concentration of DNA floating around in three-dimensional space, it's harder for the pore to grab hold of it because the pore is locked on that plane, two-dimensional membrane that it floats around on. And so in order for it to grab a piece of DNA, it wants it in that plane. And so if you tether the DNA to that membrane, it's easier for it to grab hold of it and suck it through effectively. It doesn't suck it through, it gets pulled through by the charge. But does that make sense to everybody? So effectively you increase the concentration by pulling those molecules down onto the membrane, and pore can actually sample them. What they also do is it reads both strands of the DNA molecule, and it achieves that by ligating one of the ends of the DNA up. So when it gets ligated, they insert a little marker into that ligation point, so that as the DNA gets sucked through and it gets separated, as you can see here, into one strand, and you have a little video, but I don't think it works in here. It sucks the DNA, well, it pulls it through, and it pulls one strand through, and then when it hits the junction where one strand turns back to the other strand, it reads that little encoded "I'm turning to the other strand now" sequence, and then it reads the other strand through it. So you get a read from both strands. That actually improves the fidelity of the read as well, which is quite important for a system that currently doesn't have fantastic fidelity. We'll keep going. I'm going backwards to see full. Okay, the library preparation isn't that much different to any library prep that we do in the lab. You know, you would ligate adapters. The only difference being is this linker that you add to the DNA to join the two strands together, because it sequences in both directions. Click the right way mark. He talked quite a bit about sizes and fragmentation and how long the reads were, and the average read length for what they were getting off the machine at the time of the conference was about 8 kilobases. But the reason for that is not because it can't do much longer reads than that. In fact, the record while we were at the conference was up to 50 kilobases for a single read out of a MinION, and I think it's now up to about 75 kilobases. And the actual limiting factor is actually the preparation method that how you prepare your DNA. Most prep methods will shear your DNA up to about 8, 10 kb size anyway, so you're not likely to get too many reads that are bigger than that with current preparation methods. And he was really sort of highlighting that there is no limit to how much it can pull through. It's really limited by the length of the fragmentation of the DNA. He talked a little bit about the workflows, and basically, you know, this thing runs in sort of 1 to 3 hours depending on what you want to capture. But you know, people are running them for days continuously, and they seem to be fine. And they're also reloading them and running them, and they seem to be running fine too. So this issue of base call inaccuracy, as I said, you get a current that comes out depending on what's actually sitting in the pore at any one time, and then there's got to be a way of interpreting that, and that is where they've had all the difficulty. Basically, this just sort of goes through different waveforms. This is actually what it looks like, and then they interpret the signal, and then they use these maps, effectively like digital signatures, to then map to known sequences that they've put through the MinION already, and they know the sequence of, and they're trying to match these waves to known sequences, which is where, as I say, most of the difficulty is coming. This talks about 1 and 2D data going through both strands and how going from one strand to the other improves the fidelity. What was this slide? Again, some more squiggle plots. Really much there. Errors. We're getting on to errors now. People always, one of the big things about sequencing is always, well, what's your raw read accuracy? Now, the instrument I talked about right at the start, the PacBio, has a raw read accuracy of around about 85 to 88%. So that means more or less 15 to 20% of the time it actually gets the wrong answer for a base. And then you think, oh my God, that's terrible. Like, how am I ever going to see something like that? But the trick that the PacBio does is it actually circularizes the piece of DNA you're sequencing, and it rolls around it over and over and over and over again, like hundreds of times. And so that error, which is fairly random error in the PacBio, gets ironed out. So you end up with a a cumulative read accuracy of about 99.99%. With this system, you can't roll around the sequence, so you are very limited to what its raw read accuracy rate is. And at the moment, it was running at around about 87 to 88%, which sounds pretty bad. But most of that error is actually uninterpretable word sizes, and that's to do with the library that they have for the sequence and what they're actually measuring in the current. So it's a bit like trying to match, data to a huge table of information, and they just haven't got a match yet for that particular signal. So I think that will improve over time, and most of the errors that you see in the MinION data are actually gaps. And the reason for that is because when it misses something, it usually misses the whole set of 6 because it just can't read that word. It doesn't know what word it is that is in the pore. It doesn't know which set of 4 or 6 bases is in there, and it says, I don't know, throw it away. And so you end up with these gaps, that are usually somewhere between 3 and 6 bases in the reads. The good thing about that is, because it reads both strands, when it goes back to read the other strand, it usually corrects that. So although the raw read for one strand is 85%, by the time you get to 2, it's well over 95%, which is much better. And I think that's going to rapidly improve over time. I have to say there were a lot of people, I reckon the audience was probably split into the skeptics and the believers. And then there were, on either side of that, there were the skeptics that were saying this can never be done and it's just heresy that you're ever going to even trying. And then on the believers, there was the, this is God, God is all great. It was really quite that dramatic. And when question time came at the end, it was the two spectrums right at the ends that were going at each other like you wouldn't believe. And then there were, of course, the Illumina people and the Iron Torrent people who were coming in saying, no, we're still good, listen to us. It was really interesting and quite dynamic and quite heated in places too. So there were a couple of presentations from other people who were presenting MinION data, but and the problem with getting the information from the actual manufacturer is that they're always going to present what you think is their best data. He was constantly saying that this isn't the best data we get. He said I purposely tried to get something that was run of the mill. And to be fair to him, I think he probably did, because the other presentations that presented data actually had data that was very much similar to this. So he probably did not pick their best data to actually show. But you can see here how they try and call the different bases based on these patterns. God knows how it works. I'm sure it's all proprietary. And basically it just interprets it and pumps out the quality score for each basis. So they've used E. coli as a data set, and they found a consensus accuracy at Q50, 99, and that's basically sequencing it, using the MinION process. That doesn't mean they're anything to do with their raw read accuracy or anything. It just means that when they tried to sequence an E. coli genome using this device, running it for however long they needed to run it for, they could get 99.999% accuracy over the entire genome of E. coli.
Did the Nanopore, this MinION test, they got a Q10 quality score, Q10.
For what though? For how much?
I don't know, but this was, they were part of the pilot testing, and most of their data was Q10.
Yeah. When was that though?
That's what they were talking about.
Yesterday. Oh. It's funny because there was a presentation on just a little bit in the afternoon session from Iron Torrent, and basically he was saying that this can't be right. This data, basically he was saying, could not possibly be right, because he's seen data from MinION and it's nowhere near anything like that. but then the talks that came afterwards with actually presenting real data proved that actually you can achieve those values.
It probably depends on how many times you run it.
Well, it does. It depends on how long you run it, how much time you run it, how much of the same molecule you've got in there, what coverage you've got. And exactly. You know, if you don't run it for long enough, fine.
If you want to get it to that, then you will do it. If you don't know, sorry? No, I don't think it's competitors. It's, yeah, that's why. It is at a stage where if you don't know what you're sequencing, it's going to be hard to be in the thing to say you are using the data. You know, if you know what the sequence you put in and you know how long you need to sequence it to get it to that, then it's fine. I think that's where the problem is with any nanopore at the moment. That's why it's not out. So you need to be keeping that in mind. It's not about saying this is crap, it's about that's the challenge. That is the challenge.
Okay, I've skipped over a few slides which really were talking about interpretation of signal and all that stuff to go through what the throughput of the devices are. And, you know, like you said, the throughput is really dependent on the number of pores, the speed with which it goes through, and basically the time that you run it for. And actually the figures that they had, and I think there might be something coming up in a minute where they put lots of min-ions together and made this device called the Prometheon, which is many min-ions together, and the throughput was just staggering. It made you know, the HiSeq 2000 look like, you know, good old dye termination sequencing on autorats. You know, it was stupid amounts. And you can see here, you know, that at the moment we're down in this sort of region, producing, you know, around about the 5 sort of gig sort of range of data up to, you know, maybe 10, 20, maybe 30. But you know, their proposal is to move into this range here fairly quickly with the future products, the Prometheon and everything else. This was just showing how reliable the channels were. So if you have an active pore on the min-ion device, that it pretty much stayed active for reasonable lengths of time. It ran it over 4 hours here, but there were other people that were running it for days and didn't seem to show any loss of reads from active wells. Read lengths, again that's the distribution of read lengths that you're seeing out of the pore. As I said, it's sort of around about that 8 kb mark is where they were sitting. Lambda reads. So that they're constantly updating their processes, as all these kind of sequences are over time, but particularly the ones that are in their infancy, as they realize where their difficulties are and their bottlenecks in whatever they're doing. They are constantly changing their chemistry. They're actually looking at changing pores, and they're looking at changing those motor proteins, which are the ones that we talked about that are actually breaking the DNA as it's going through. And by breaking, I mean slowing it down, not actually cutting it. And they found that now they're moving from R7 to R6, sorry, from R6 to R7, they're getting much better signal to noise, basically, is what they, you know, you're always going to get a level of noise within any electronic system, and they're finding with the new chemistry, the R7's much better than the R6, and that I think is what we will have if we get stuff from min-ion to actually do our own work. We will get the R7 chemistry rather than the R6, because they've pretty much changed that now.
What is it that is not right now?
R6. But all the burnings are done with R6, all that, but the new stuff is all coming out with R7. And you notice that it evens out the strands as well, where with R6 for some reason you have a lot more noise with the complementary strand. Didn't really say why that might be, but again, And now they're working towards R8, and again, the R8's even looking even better. So, there is a lot happening there. There's a lot going on, and I think there's a lot of promise in the technology. Whether these biological pores remain or whether they go for synthetic ones that are easier to control and, I guess, less prone to biological degradation over time, I don't know. But certainly the technology changes in the pores and the membranes are really what they are focusing on to a certain extent. MinION Access Program is what we are now in. A lot of information on the website now. There's a good network of people that can exchange ideas and talk about problems that people are having with the actual sequencer or their technology or their particular type of sequencing or their DNA or whatever it is that they're actually doing with the MinION. So it's gathering momentum amongst the community. And there's a small piece. This is actually their stockpile of MinIONs, and you can see all of them here. It's actually quite a lot. As I said, there are lots of, they talked about upgrades and what they were changing and kits and how they were basically, everything would be coming out in simple kit form, most of which wouldn't need, crazy stupid storage at -80. Software is all available online. Don't recall what Skunkworks was, but did it say there?
The MinION software is also updated.
Yeah, it is very frequently updated, and that's again, it's largely because of this issue with increasing their library word size. So as that dictionary increases, they're able to call things more accurately, interpret things in a different way or more accurately. So I think that's going to happen for a while, yeah, I suspect. So this is and then those of you who've seen the current MinION will know what it looks like. This is the next MinION release, which is cooled. It can actually run a lot faster. So I think, again, they've made some more changes to the ASIC inside and some changes to the pores and membranes to help things run a bit quicker. This is the thing I talked about earlier, which is the Prometheon, which is basically a mass of parallel MinIONs all in one device. Again, it runs on the same principle. No different. You just load your samples up and you run it for as long as you want to collect the amount of data that you want. Yeah. So 25 to 100,000 channels. That's basically a lot of information that's coming off. This one you can't hook up to a laptop. This one has to go onto something a bit more significant. So again, this is where Promethean's going to be, up in this range, which is outstripping the HiSeqs and the other big players in the game at the moment. One of the things they're particularly interested in, as all I think of these DNA sequencing people are, is sample preparation. How easy can we make sample preparation from, in order to get the person from their sample to their data? And really they're talking about, literally spotting a drop of blood onto a device, and then basically that gets loaded straight into the sequencer. And so your human genome will be sequenced from a spot of blood. It's like Gattaca, isn't it? Have anybody seen that movie, Gattaca? Unvalid, spot of blood, bang, you're done. It's coming to that sort of level. And one of the presentations that was given by somebody from Birmingham University later on in the day, he likened all this sort of, he was in the I Love MinION camp, I'll just declare that, but he was likening it to, Star Trek. He must have been a very secret Trekkie, I think. And he was talking about tricorders, and how this is a bit like a tricorder, and it would be able to detect somebody's DNA just from them touching the... He was off. He was excited, bless him. So, this is not just, this idea is not just something they're thinking about. It's actually happening, and these are the the new devices that will have this spot-on, what they call technology, where the blood gets spotted on and it just runs through, gets separated out and runs straight into the machine. And of course, this was a question that was asked earlier in the day before this presentation was put on, and that was, what about other nucleic acids? What about RNA? What about all these things? And basically they said, yep, we're already onto it. So they are indeed already onto it, and he did have some information that after the presentation was given about how far they've got with this. There wasn't too much in the slides, I don't think. That's pretty much it. Obviously it has a huge, the spot-on technology has a massive range of things that it could be applicable to, and particularly when you're going out into the field, for example. If you're just taking a laptop out and spotting your favourite piece of whatever it is onto the thing and getting sequence data out, it has great application out in the field. So they're talking about how that might benefit, biosecurity, foods, waters, all these sorts of things where they might need that sort of information. So that's about it. As I said, there were some other technologies that were talked about in later talks, and I'll give you an insight into those next time. But I think in the interest of time, I'll hand over to Fran. Is it on here? Do you need to load anything up? Which one? That one? Okay, I'll hand over to Fran.
Alright, so my presentation is basically just an update of what I've been doing the last couple of weeks, because I've been making these presentations and then the session's not been on or I've been away. So this is kind of an amalgamation of a bunch of presentations that I have done over a couple of weeks. So just to remind you all, my honours project is about methods for reducing sequence resampling and improving sample enrichment for human virome sequencing. And my main aim is to reduce bias present in PCR with the possible solution that digital droplet PCR and its compartmentalisation of the PCR reaction might help to reduce that bias by separating out the templates in a sample so that the same template can't get amplified over and over and over again. So we don't see that bias at the end. So what's happened so far, I've been able to recover sequenceable product from digital droplet emulsions after digital droplet cycling. However, at the moment it's a bit unclear whether aspects of the digital droplet emulsion reaction might be inhibiting my reaction.
Which reaction? The sequencing or that?
Either the sequencing or just during the PCR. It's hard to work it out at the moment. So I'm seeing changes at the sequencing level and changes in the product straight after digital droplet. So what I did a while ago was I took a HCV sample that had been primary ID tagged a number of times, so I knew that it could be done successfully with this sample. Put it through cDNA conversion, tagged it with primary IDs, had 2 replicate cDNA samples that were both tagged with the same primary ID primer, put one lot of that cDNA through digital droplet first round amplification and one through normal PCR amplification. With the idea then that at second round I would put the digital droplet one through both PCR and digital, and the PCR one through digital and PCR. So I end up with 4 different options at the end, which I would then take through to sequencing and compare. However, what happened was I could not read anything with my digital droplet reader from the first round or from the second round. And the second round digital droplet products, that had been through first round digital as well. I couldn't see anything on a gel, I couldn't quantify it at all, there didn't seem to be anything there. Although I would note that first round digital droplet product, you could see something on a gel. So there was DNA there was just something going on with it. So, yep.
Have you tried sequencing just the first round product, whether it's a correct one from the droplet as well? Is that possible?
I didn't end up sequencing from first round, but I did take that first round digital droplet through second round conventional PCR, and I sequenced that. So it was the right product. However, it was a bit, there was a bit of a mixed signal. And I'll go on to that. Yeah, there was a bunch of things that kind of went a bit wrong, and I figured some of them out, but not all of them. So I found an issue with my primer sets with the digital droplet. This first time I went through it, it wouldn't work with my second primer set, which worked with all my PCR versions of the same product. And the sequencing output, so I sequenced the digital droplet second round that worked with one primer set, along with my just PCR second round product, and a positive control, which was from the same sample that John had taken through to first round previously. So what I got is a good sequence output from John's and quite poor from mine, which I had put down to a problem with the cDNA primer. So I had freeze-thawed the cDNA primer probably a few too many times, and that might have made it a bit inefficient at tagging and changing the RNA. So also what I observed was these little intense peaks down there are the primer ID tag, and you can see there's quite a lot of primer ID tags going on in John's sample, the control. However, in mine, you can't even really tell that that's primer ID tags. So it appears that there's like a lot less primer ID tagging that went on in my products here, which if there was degradation of the cDNA primer, that could be a reason for that. So then I went and tested the digital droplet supermix. So without generating an emulsion, I just took the supermix through conventional PCR to see if I could get things amplifying properly with that. So here I used John's first round product just to control for anything that might have been happening at the front end with my RNA extraction and my cDNA conversion. So I knew that this sample had gone through that all right. This was my positive, so this is PCR mix and the PCR cycling conditions. These other two were the digital droplet mix, and I tested cycling conditions as well to see if there was any difference with the standard digital droplet cycling conditions, see if that was a bit more or a bit less effective. But what I found was that there was really no difference in cycling conditions. There was really no difference in intensity of the product that I got from normal PCR and from the digital mix. So then I repeated the digital droplet PCR with John's first round product. What I found from that was that I could break the droplets and get a fragment out of it from John's. It was a lot less bright than the fragment that I would get from second round PCR, where essentially all the conditions are being kept the same. It's just the only difference here is that I put one lot of samples, my dilution set, through an emulsion. It was the same super mix, the same everything. So obviously something about that emulsion generating is doing something to the sample.
So this is your recovered, right?
Yeah, this is my recovered one.
Could be losing something in the recovered.
Could be, but then that wouldn't explain my next slide. So I might be losing something in my recovery process, However, I would still expect to see some strong positives on the droplet reader because I'm taking a first round product through second round, so surely a lot of stuff should be coming out, especially if I'm seeing something on a gel. But with the droplet reader, I mean this first one here should be my most concentrated sample, and it's coming up just looking as a plain old negative. That's the end of my negative there. That one is my 1 in 100 dilution, and then my 1 in 1,000 dilution comes up with sort of a bit of noise that maybe is a positive, although that's the opposite of what I saw on the gel. So on the gel I got a clear band from the most concentrated product and less from the others. So it's all a bit all over the place. With sequencing, so I just finished doing that today of the product that I just broke from the droplets, and that was from John's first round one. So what I found there was that the forward primer sequence seemed to have worked really well, although with the digital droplet sample there appears to be less primer ID tags than there is from the PCR control. But the reverse sequence for the digital droplet looks very messy, like a very mixed sequence, although my control from just the PCR method, the reverse sequence looks fine. And there was, yeah, just lower fluorescence overall for the digital droplet, possibly because of less product there, bit of mixed reads. So in summary, I have checked the digital droplet reaction mix, the cycling conditions, checked for any issues up front with RNA extraction or cDNA conversion. Currently considering the possibility that there might be inhibitors present that are somehow, you know, producing less product in the digital droplet emulsions but not in normal PCR. I've checked for contaminants, no contaminants anymore. So my question would be, why are PID products retrieved from digital droplet emulsions appearing less abundant and with fewer PID tags? So there might be loss of DNA through the droplet breaking procedure, but why then am I not seeing anything on the droplet reader? Because obviously nothing's being lost from there. It's working when I do it with PCR. I'm getting a good droplet count, so event count is quite high. So that could be due to the presence of inhibitors, or I guess it's possible that less sequence resampling is occurring in the digital droplet and therefore less product is coming out at the end. Yeah. Thank you for listening.
Inhibitors, Fran, you won't see recovering, you recover from the droplets.
Yeah.
Still, I cannot understand why when you recover, you're seeing a fragment in the DNA. You recover from the neat sample.
Yeah.
But your neat sample is not showing the droplets there. The only explanation I can give there is your negative is not really a negative there. All of the samples are positive in the digital. You know, you're using the duplicate of the same thing, and you've recovered a product from that one, from the meat, right? Yeah. You've recovered from the meat a fragment. So if it was inhibition, you wouldn't recover anything from that.
Okay.
You wouldn't recover anything from that if there was inhibition. So I can't see there's any, inhibition of the PCR. Yes, it could be, if you're recovering, you would lose some amount there, which is fine. If you change, you're picking up only a certain amount there. If the whole DNA from your, whatever you're using for spinning it out from the oil, DNA is not recovering, yes, you would expect less recovery. But the fact that what you're seeing, the first one, which is meant to be a neat sample, you're seeing a band recovered from there and very little from the other one. right? Nance's thing is that all these three are positives. That's
Although it does seem like quite low amplitude from my understanding for positives, isn't it? No?
30,000 is not low.
It's a very high background.
Okay.
30,000 is very high. 30,000 is not low.
The bit where you can see a little bit in the corner, it's not run
See,
properly.
that's why that's a broken droplet.
Mhm.
That's negative. That's not really a true negative. That's a broken droplet, right? So I wouldn't even consider that as your negative background. So the only way you can say what you've recovered from it is that all these are actually positives. And 30,000 is quite high. In evergreen, 30,000 is probably the positives, right? You normally get a background of around 20,000. 15,000 to 20,000 is the background.
Okay. And then 30 is positive.
30
Right.
is positive. So all our other assays which we do on the digital, 15 to 20 is normally what you would see the background. And 30 you will definitely see them as positive. So, so one is thinking that all of these are positive, and that's the most concentrated. That's why you see the recovery from there, but others may not be as, and you're not seeing it on the gel.
Yeah, okay.
That's my only explanation. Otherwise, I cannot explain. They've gone through the same cycling conditions, they've gone through the same process. Why are you seeing it on a gel? If it was inhibition, you would not see it on both of these.
Yeah, all right.
Yeah. What would inhibit is all this thing is doing is reading it, right? All it's reading is the fluorescence.
Mm-hmm.
So it's not making it do anything different. So I cannot.
So then it would probably, if that is all positive, it would just come down to some aspect of the droplet breaking procedure as to why the sequence from the reverse primer is
So it's
a bit ugly.
inhibition in sequencing.
Why would they be all positive? There's no negative droplets there. Where's the negative droplets?
But if you're putting in, this is primer ID first round product. If you're putting in 20,000 copies, right? More than 20,000 copies. These are 20,000 droplets. Everything will be positive.
No, you need to put a ton more than 20,000 copies to get.
Yeah, but it's more than 20,000. See, but 1 in 10 and 1 in 100, you're starting to see those.
Where?
So these are 3 different dilutions. We've got neat, 1 in 10 and 1 in 100 dilutions.
So it's a 1 in 100
These are
here
different
and 1 in
Oh,
1,000
I see the
dilution.
Yellow line. I
Yeah.
can't see that. That's a terrible.
Yeah, that's-
colour.
Yellow.
Yeah, but where's the negatives and the positives then? Am I missing something? Aren't the negatives supposed to be down the bottom?
See, the fluorescent with evergreen, normally, yes, see, you need to have a negative control. So the negative control didn't work very well. So A4 is meant to be a negative control.
So the far left,
The
where
far
it is
left.
that just fraction higher,
Yes.
that's the negative.
But that one didn't work, that's always the thing of that first well doesn't work very well. So you need to have at least 2
Two negatives.
we said before that you need to have
How does a negative control become fluorescent then?
It's not fluorescent. Here nothing is being analyzed, so you're seeing everything as black. If you put, manually put in, you will see the blue color.
No, I'm worried about
No,
colours.
but
I'm just
what I'm saying is if you see the same thing as histogram, you can clearly see there are 2 different populations in your C4 and D4,
Yeah,
right?
yeah, because I could draw a line here and then that would call it
Here
2
you
different
can see
things.
2 different populations of persons. Yes, there's not, clear segregation, but you can clearly see that. And
So you're saying that band that's just below 3,000 for negatives?
30,000.
that's
Sorry,
30,000.
30,000, yeah, 30,000.
But could be, but I don't know if it is, because when Francisca is recovered the DNA by breaking the emulsions for the duplicates of these same samples, right? So the first one, she's recovered DNA from this. So she set it up in duplicate. One set to do it in the reader. So this bit, so these are 3 different dilutions, right? This one, when she broke the duplicate of this one and recovered the DNA, that's what she showed in the previous slide.
Yeah,
that
that
when she got
is
this
here.
one.
So
That's from the recovery of that particular sample.
hmm And then the next
so
one
that's
is the
come
one
from here, the first web. So that seems to suggest that it's the right size and it's positive. It's duplicate, exact duplicate of that same one. So you have recovered a product from there. Okay, so
So
that
that
seems
there
to
is not
tell me,
negative.
that seems to tell me, just go back to your digital. So this tells me that this is actually positive, it's not negative, that every droplet here
So what's that other thing at the end then?
Yeah, so here there's sort of 2 different proportions of 2 different populations, which is you're having high fluorescence. So when you're diluting, so there are 2 things. One at a high, which is what we see when we do a second round nested PCR from a neat sample. You start getting inhibition at the neat sample, or you start seeing the products don't work very well.
But
But when you dilute it,
the duplicate worked
but
perfectly.
the duplicate worked perfectly, but it's, some of the droplets may have worked and you recovered it. But the recovery is less, so you don't know whether the recovery is because you're not recovering the full amount percentage of DNA that is there.
I think until I see a negative control that's properly negative.
Exactly, you can't say anything. But because she's recovered the thing and it's got the size, that tells me that this has to have some positivity.
And that doesn't, it just tells you it's a duplicate, it may not have worked.
No, it's done at the same time, unless hasn't added DNA in it. She said she was very careful and she added it. You have to have faith in
2 positive, 2 negative controls in each run.
Yeah.
Have you had the negative control, or which was a split droplet, or however you call it, also on the gel? Did you do
Yeah.
the other part on the gel?
The other negative was a negative. She had
This one.
Oh,
No,
one
no,
on the
no.
left, first one, or A, or
That one?
or one. Yeah.
Yeah, that was run on the gel. That was my negative.
Have you ever seen
So, but it has got a slightly lower band down
Yeah,
there.
that's prime down.
Yeah, but still, it was still correcting your recovery
Sure.
or however you
Oh,
call
okay.
it, so it hasn't necessarily going to have no losses at all, as I understand it. I mean, so if you have lots of primer dimer in that sample,
Mm.
that might create a high background to my understanding, isn't it?
Yeah,
Sure,
yeah.
That's
If you put
why
an
I
extra
agree.
primer, maybe that actually improves your results.
with the private government small, so they process large?
No,
It
not
depends on how
necessarily.
much, I mean, just because the band is lower doesn't mean you can't have a lot of fluorescence from a small product, as I understand it.
No, no, no.
Yeah,
but
but
the
it's-
fluorescence will be different. And primers will be less, and that's why the background most of the time is between 15 to 20,000.
I've never done a droplet, so it's just like,
It's a very easy hit, isn't it? It's totally-
And how many wells are positive too. So all those things will make a difference. So yes, the primer dimers will have a fluorescence, and that's why the background is there, because evergreen will bind to primer dimers, and you will see that.
Yep.
And possibly that's what we are seeing, but when she's recovered it, you're seeing the size, which is an expected size of the band that you expect around 500 base points.
So it's- it's been worked, is that you could have like long
Yeah, it's
lines
like
or
size
just
of
one
these.
set, yeah.
It's
So-
like size of
So the process would be proportional to the length and the number of
Yes.
copies.
and you will still have your CDNA product in there as well, as I understand, isn't it?
This is a first round product.
But you would still have your template in there, if you do a first round reaction, you
Yes.
still have your template in there.
Yeah, but
And you would still bind to that one as well.
It won't, so the single strand.
But it's not, it's first round.
You have a CDNA
Yeah.
conversion, then you have a double-stranded CDNA or however you call it,
Yeah.
and then you do a first round. So
Yeah,
you
yeah.
still have the
But
original
remember, if you're
product.
putting in only 20,000 copies, and the amplification, the signal is going to be very low for that.
Mm.
Yeah,
How much
for your
templates
first
do I
round
need to
product.
do?
It's amplification which increases your processing. It's like any real-time app.
Yeah, but it's still telling you how much, what we're doing next week, things to do.
Cool.
Product, it's not notable.
I'll go in
If you have a lot in there, you would get a higher value.
Sure, sure.
So how much DNA is going into these reactions?
So this is where she did 3 different dilutions. So I don't know what your first round is like, nothing, right? Your first round amount is, you don't see it on the gel, so it's very low amount, right? But she's done 3 different dilutions of
All I'm
that.
saying is that it's less than 5 nanograms per microliter. So
Exactly, whatever
that's
the
still
liquid-
billions of copies.
But millions of copies, but 1 in 100 dilutions is still, you've
It's
done
still
it
hundreds
in
of
range,
millions.
and she's not seeing any of that recovered on the gel from there. So even if you started with that,
We're
you
nowhere,
should see it from.
we're nowhere near with the range of 20,000 copies.
Sorry?
We're nowhere near the target of 20,000 copies.
No, but you're not still recovering it. You should see everything positive. If you had 100,000 copies, you should see everything.